researcher at the Karolinska Institutet in the Division of Genome Biology and Department of Medical Biochemistry and Biophysics. He received his bachelor's in biochemistry at the University of Tübingen, Germany in 2007, went on to do his PhD in Rockefeller University under the supervision of David Alice, where he investigated the biochemistry structure and function of histone variant H3.3 and its cognate histone chaperones. He held Boehringer Ingelheim Fond's PhD fellowship. He had lots of fellowships along the way and graduated in 2012. He moved to the MRC Laboratory of Molecular Biology in Cambridge, where he was working as an EMBO fellow and several other fellows, it looks like, along the way, working with Jason Chin. He has published impactful studies at every stage along his career. Since 2015, he's been an associate professor at the Karolinska Institutet in Sweden. His laboratory develops new synthetic and chemical biology methods to perform biochemistry in living cells. The aim is to understand protein function in a mechanistic and quantitative manner in the context of living cells. And so we're really excited to hear what you've been up to. Take it away, Simon.
Well, thank you so much for the introduction. And I thought I'd focus on tools today rather than biology, and I hope that will cater to the audience because I think in the GCE field we are often thinking about how can we get the chemistry in, and then one thing is doing the chemistry and the other thing is getting it to work in a cell that you like. So we are working on the second part, really complementary to new chemistry. And of course, everyone here, we're excited about the prospect of augmenting proteins in living cells, specifically in mammalian cells, with non-canonical building blocks to encode for new function. I think we are all aware of the challenges. I will go into a bit more detail. The fact that in mammalian cells, at least, we don't have the luxury of creating codons. We have to work with what we have, and we have to recode existing codons, and that creates always a somewhat incomplete solution. In this case, amber suppression, suppressing the least abundant stop codon, the amber codon. We need a tRNA that is complementary against the amber codon, and a synthetase, cognate synthetase that can charge it with a non-canonical amino acid. And so in principle, that's simple, but in practice, of course, there's some key challenges here. How do we really ensure that we have selective recoding the amber codon where we want, and efficient recoding, but not necessarily recoding endogenous stop codons? And going to get to that a little more, but just what does the literature tell us about this problem? It's always kind of a miracle in the field, so to speak. Why does it even work if we have 22.6% amber codons in the human genome? Why can we recode anything specifically? So I think we are quite in a lucky position, because nature has sort of encoded termination of translation in different ways than just a single stop codon. It's the context that matters as well, and that gives us some window of opportunity where we can get efficient recoding of a premature stop codon in our protein of interest versus proper termination of endogenous amber codons, as well as other processes like mitigation of elongated proteins based on decrons that happen after the stop codon. So we've been basically trying to, in many ways, both apply genetic code expansion in mammalian cells for biology, but also really drive the technology and the ease of use and the robustness, and also the universality of the approach, particular to cell lines that we care about, which are typically not a HEC293 T cell. And we've been doing various assays like microscopy, proteomics, and also we are interested in, basically, as David showed, some interesting applications in studying the histone code with post-translational modifications, but I will not talk about much about those today. So I'll talk about stable cell lines that we generate, because it's rarely done in the field, and I just think it's kind of interesting to capture a little bit what can be done and also what can't be done. So 98%, I guess, of the studies are done in transient transfected Hector entry T cells or COS7 cells, and that creates a heterogeneous pool, and it's useful for some application, but of course we all know it has some limitations in reproducing results over different experiments, because we have a transfection efficiency as a confounding variable. And a little is known about like how the cells do, essentially, if there are any like off targets to the specific technology that we're interested in. So one of our aims is to really understand systematically what's really limiting efficiency versus selectivity in a mammalian cell. And we've been working with a plasmid design that's quite universal. In Jason Jin's lab back, I wanted to create a system that I can integrate. So these plasmids that we use, they have these red terminal repeats that allow integration with piggyback transposase. And even though they were intended for making stable cell lines, we realized that they were also very good for transient transfection and extremely efficient in supporting amber suppression in transient system. Here with Wolfgang Schmid, we first used them to really show like they can push the efficiency sort of towards 100% incorporation rate of non-canonical amino acids and protein. And all the subsequent plasmids in my lab, they are available on AdGene. So if you have any use for them, get them, and also just get in touch if you have questions about these reagents. And we accumulated quite a toolbox. It's always modular because we separate the synthetase on one plasmid from the protein of interest on another one, simply because then you can often mix and match the plasmids in different ways to achieve different amino acid incorporations, for example. And each of the plasmids has 4 copies of tRNA, and you will see later we knew from prior studies that was important, but all our results also support the notion that the tRNA levels are really what's limiting amber suppression in mammalian cells. And then we can, do transient transfection or make stable cell lines. And if there is a need for defined integration, we've also developed a protocol to integrate these plasmids out of the box using CRISPR-mediated non-homologous end joining. So we linearize the plasmid either by a single cut or 2 cuts in situ after transfection with a guide RNA pair that one targets the plasmid, one targets the target locus, like the safe harbor locus AVS in human cells. And then we can show that it can also generate defined integration sites. But for the next study, really, we've used piggyback integration. It's quick, and it also allows for potentially multiple genomic integrations. We don't often know the exact number, but where we have looked from what we know, I would say that it's between 1 and 10 integrations using piggyback. I don't think we ever get more than that. So we have potentially, even though it's from an integrated locus, we have potentially high expression because we have multiple copies in the host genome incorporated. And we simply wanted to ask Birte, a postdoc, and Johannes Heimgärtner, he was a master student, now a PhD student in Katrin Lang's lab. How well will it work in different cell lines that we are interested in, for example, cancer cell lines? And so in each case, we transfect those with 2 plasmid synthetase and a GFP reporter with an stop codon. And then we do plus minus amino acid here, cyclopropene for bioorthogonal labeling. And then we both check the protein level, but we also label the lysate. And even though now these are stable cell lines, you can see that the lysate labeling really shows that the amino acid is quite selectively incorporated in the target GFP, and particularly well in HCT116 cells. We never really able to figure out why some cell lines are more susceptible. Are they actually less stringent in their termination, or do they express much higher level of tRNA? or the target gene. We don't really know that at this point, but it's empirical observation that some cells like HCD116 colon cancer cell line are particularly well in creating stable AMBER suppression. To the extent that when we isolate these stable cell lines, you can see that in the case of HCD116, extremely high expressing. And if you sort of look at the total protein produced, it actually rivals a transient transfection in HEK cells. So for protein production purposes, if there's anyone amongst you guys that is interested in producing engineered protein with non-canonical amino acids, I mean, HCD116 cells seem to be a very good host to do genetic code expansion and protein production in a stable cell line. Now, there's also something that we can test here, especially in those cell lines that are not so good. You know, why are they not so good? What's missing here? So we try complementation. In these HEK293 T cells, they are mediocre, basically. Can we make them better? So we essentially add synthetase or tRNA, and clearly synthetase addition doesn't really help, but tRNA transiently transfected and will increase the read-through of the GFP. That's all integrated. So this is a clear effect of recoding efficiency increase. And then once you have more tRNA, synthetase seems to have some additive effect on top of that. The nature of the tRNA doesn't seem to matter. We've tested different variants like the N15 from Irene Cohen's lab, which she showed is more stable, and the A21 from Abhishek Chatterjee's lab, which is also shown to be more efficient, but in our system, they didn't bring an advantage, and neither did a more catalytically active pyrolysyl synthetase. So there's sort of a hard limit, unfortunately, in cell lines that you will never reach the levels of a transient transfection. And transient transfection, you simply have hundreds, if not 1,000 copies of your transgene. So clearly that is a different regime on every level. But you know, something that we at some point wondered, like, is there a real reason, or is it simply a historic artifact that everyone is using AMBER? Because maybe, you know, this was the most the one that was historically used in E. coli, then used in mammalian cells. It is the least abundant, but maybe it would be better to use a different stop codon. Ultimately, you can see here that we didn't outsmart the rest of the field. No, I mean, AMBER codon is historically and empirically the best one to use. And you know why? Again, it's very interesting that TGA is known to be the leakiest codon when it comes to endogenous read-through, but it's not possible to suppress it efficiently or as efficiently as the AMBER with a pyrolysyl system, at least. While this doesn't really help us make better cell lines per se, obviously changing the AMBER codon to another subcodon will allow us to do multiple amino acid incorporation. And for that, we also need orthogonal synthetase pairs. And so we use, we generated another plasmid series, which has the tyrosyl or the leucine tRNA synthetase pairs from E. coli incorporated. Originally described by Sakmar, Schulz, and Chatterjee labs, and those you know, have been shown to work very well in mammalian cells, and they also do when we integrate them with piggyback transposase. And again, like some are really like striking, like acetophenylalanine. If we sort single clones, we can be extremely high expressing clones, again in a HCD116 background. Now, is that, you know, really useful? I mean, there's a lot of applications then in you know, looking in more relevant cell line than HEK293 T cells, and even in primary cells, we have shown that, you know, in principle, you can make a genetically expanded code cell lines in the background of human iPS cells, and you can generate, you know, complex organoids which support incorporation of a non-canonical amino acid. In this case, you know, in GFP, And interestingly, in this brain organoids, not every cell seems to do AMPA suppression to the same efficiency. It seems here neurons are doing particularly well, and that's why we kind of label the neurons and the organoids specifically, but that was not really intentional. That just is an interesting biological feature, I have to admit. And now you will ask, but no one has something useful to do with GFP. So what's with real proteins? We just did a series of proteins that are interested in, or that are kind of representative of different, use of fields. So one is obviously receptors on the cell surface. G protein coupled receptors here, CRFR1. Now that we generate in a stable cell line, we select the cells after piggyback transposition, and with a cell impermeable dye, we can get very nice labeling on the cell membrane. I don't show the controls here, but of course it is specific to the cell line that expresses the receptor on the surface. And more importantly, when we run this on a gel, we can also show that essentially with the cell impermeable Alexa 488 dye, we don't have any other bands that are labeled. So it's really selective for the receptor, also in the case of another receptor, SYNOCH. But as soon as we're using a cell permeable dye, you can see that really many other bands take over, which will be residing proteins in the cytosol, endogenous proteins that essentially get mislabeled by the non-canonical amino acid. And that really shows that when we go to a stable setting, we cannot really rely on this absolute specificity, and GFP in this case was kind of an outlier that is simply so robustly expressed and stable and accumulates at high levels that it can be selectively labeled, but that's not true for many endogenous proteins. In the paper, which is in press at Cell Reports Methods, we show another few proteins that also don't work. So intracellular labeling here, for example, protein DAX and tubulin. You can see like the tubular structure with the HA tag, and you can guess it in the fluorescent label channel with methyltetracine, body PFL, cell permeable, but it's not usable at this point. And so quick summary here. So stable cell lines, if you are interested in cell surface receptors, is a fantastic possibility to use genetic code and really characterize a cell line, because you can see here that every cell expresses the receptor, and it's at a quite homogeneous level, very different than a transient transfection. But if you're working intracellularly, don't think we have the problem solved. I could talk hours about all the things we have tried, orthogonal organelles, a la Lemke's approach, and other possibilities. One day I think someone will make it work, but we haven't really found the magic bullet there yet. In terms of more tools, so one opportunity now is that we have different stop codons, and we can definitely recode also the non-AMBER codons if we want to, albeit at lower efficiency. Can we use that to do combinatorial labeling, do double labeling at two specific sites? So in this case, to generate this in a stable system, we use the what I've shown you before, the very efficient acetophenylalanine synthetase together with a pyrolysyl synthetase with a ochre stop codon complementary tRNA. And then in this case, we produce GFP for mass spec. We can show that it has the two amino acids incorporated, and you really only make GFP in the presence of both amino acids. So these two pairs are mutually orthogonal in mammalian cell. That's of course known, but it also works in a stable setting, which is quite amazing, I think, given that we really need to recode two stop codons in the cell. And each of them is not that optimal, of course. And we could also show that this can be done with a membrane receptor and now live cell labeling on the membrane. This is again SYNOCH, which has ideally, we want to do a FRET measurement here to measure the force-dependent activation of notch. But here, for biophysical experiment, we don't really have the signal to noise yet. So this labeling with cyclopropene and acidophenylalanine was quite difficult, and partly because the A side on acidophenylalanine is not very reactive. We have actually shown that in another study where we compare different A side bearing non-canonical amino acids, acidophenylalanine here on the right. In the middle is a linear A side, acetoetoxylysine. That one is really even worse labeled. And then we generated one that has this chelating group for copper, and that really labels better at lower copper concentration. So generally, I would not recommend doing click chemistry on live cells unless you're already using the bioorthogonal, the SPEEDAC reaction for another reaction. So for dual bioorthogonal labeling, obviously, click chemistry is the next best to SPEEDAC that we can really use. And with this amino acid, it was quite good, but we have to say that the best one was incorporating an alkyne and then using an A side dye. And this we have achieved using another synthetase pair that's orthogonal to the existing one. And that's by looking in the kingdom of the pyrolysyl systems. And we got initially inspired by this article that reported that there's different variants. Some of them don't have an N-terminal domain of the pyrolysyl synthetase, which was really thought to be essential for function for a long time. And then first Jason's lab in E. coli, they showed a whole range of these new variants. And by now they've shown that there's about 5 possible orthogonal pairs that could be combined in a single system. And then also we and them showed that the ALVO system initially works in mammalian cells, but that was quite inefficient. That was a little bit disappointing comparing what we had been developing in mammalian cells to the E. coli system. So then we switched to another variant initially described by Chin Lab, the G1, and we engineered the tRNA, and we ended up with a essentially quite efficient dual labeling system. This is now in transient transfections where we have the MASE, PIL-RS, PIL-T, and the G1 with an engineered PIL-T. And that's published 3 years ago, show that this is fully orthogonal, the two systems, through the engineering of the tRNA. And we can do much better dual color labeling with that system than with acidophenylalanine. Now, what I'm not showing here is one issue that we ran into, that ultimately we cannot so far generate efficient G1 stable cell lines, because this G1, I think because it lacks the N-terminal domain, it probably has a much lower affinity for tRNA, which means that as soon as tRNA levels are limiting in a stable setting, this synthetase will perform very badly. So the MASE serendipitously has this additional domain that can probably make it work with lower concentration of tRNA. With that, I just briefly show one more application, 1-2 minutes. Yes, we can do incorporation of post-translational modification, acetylation of histone. It would be great to do trimethylation, by the way. As Ben said, that's something that's not possible with genetic code, a clean trimethylated lysine in a position, but different acylations can be incorporated. And actually what's really convenient for us in that matter is that we found that between the wild type synthetase of the pyrolysyl, the MASE, and the ncAAs described by Jason Chin's lab, these two enzymes can take any of the acyls. The wild type takes the longer ones, the ncAAs takes the shorter ones. So we can actually show that we can template a single lysine, 14 of histone H3. And depending, this is like now a cell line where the synthetase is already integrated and that the histone is transfected. And depending on what amino acid we get, we get basically a different acylation on the side chain. So with this, we're going to study sort of how different acylations affect binding partners and also sort of downstream modifications. And now I have to go here, jump to the acknowledgement. First of all, we had some great collaborators in biological applications, as well as chemists that make amino acids, and a fantastic team, really already highlighted Birte Meinecke, who did all the lead development in new tools together with Hannes for a long time. Leah did the work on organoids. And with that, I'd like to thank you. Take questions.
Thank you. That was great. You're doing some heavy lifting in the cell area that I know is quite difficult. So I'll start off with a question. So out of interest in those cells that work, Ryan, would you have a question?
I do have a question. I'm very excited about your stable cell line work, and maybe that's what John's going to ask about. But also very intriguing with regard to your statements on the background encoding of non-canonicals in stable cell lines versus transient transfection. If I remember correctly, what I heard you say was you got a lot more background encoding into natural TAG codons with stable cell lines compared to transient transfections, if that's what I remember you saying, right? And I was curious, was your gene of interest also in the stable cell line or not? Was that transiently added? And is there a molecular mechanism that first, did I hear you correctly? And the second, what is the molecular mechanism for maybe that, or can you hypothesize on that? Because I think that's a really exciting.
Right. Yeah, I think, it's probably not 100% correct how I said it, because we always compare, GFP versus the background, or target protein versus the background. And in that sense, transient transfection will create a much higher level of the target protein. So if or not there is background, we don't know. We actually haven't. I mean, as you say, it could be an interesting experiment to just check the background.
Right. We also were trying to figure out, is that background that you're talking about related to the non-canonical or the RS, right? So obviously common tRNAs but have you looked at it for different non-canonicals?
So we've done mass spec, in some cases where we're pulling out, what is, we pull out our target protein, but we also pull out everything else that has a non-canonical amino acid. And what we find, and that's also been reported by Katrin Lang and Sebastian Bultmann, is like that ultimately a lot of highly expressed housekeeping proteins like in GAPDH and so on, if they have an amber codon in the organism, they will be likely to be sort of some of the main targets. And that is just sort of relative to how much we can generate of our target protein. If that ambient background becomes too large, then at least for imaging application, it's not useful. But that, of course, doesn't mean that there's not other very good applications of bioformal chemistry, for example, controlling a protein of interest. Yes, I think one always needs to then do the right control, having, in our case, we then make a control cell line that has the machinery and, for example, GFP, but not our real protein of interest. So we can compare that across this pair of cells. But yes, unfortunately, sort of intrinsically, it's very difficult to always cleanly separate when we're talking about function, to be really sure that the function that we are generating with our protein, engineered protein, is not affected by anything that could be going on in the background when we modify translation.
Thank you.
Yeah, so I have a quick question about that cell type that seems to work, the magic cell type that you're able to get really nice, stable, high expression. If you looked at the open state of the chromatin, does that just have more shots on goal for piggyback transposition insertion, or is it something else?
Yeah, interestingly, I mean, so it could be the general efficiency of an EF1 promoter, right? But then that's not really supported by you know, the PILRS level, because that's under EF1 control as well. So if it was just that EF1 promoter overshoots or the integration sites are so good in a very active context, then we should see more of everything, including PILRS. But generally the promoter is kind of the same. But I do think that ultimately the termination efficiency is a variable in biology that maybe is used for actual biology to, in some cases, you know, allow read-through of an important protein to make some alternative protein. I mean, that's a hypothesis, but or that, you know, in cancer it's simply messed up. We don't really need a function for that. We haven't checked, I should say, that's something on the list always to check actually ERF1 protein levels, which would be clearly like if that would anti-correlate, we would have a good guess.
Yeah, that's really, it was really interesting. I really